Carbs provide energy and fuel for the body.
The goal of carbohydrates is to provide fuel for the body. The different structures of carbohydrates are separated based on the number of carbons in the molecule and where the double bond is and where they form a ring. Monosaccharides are simple carbohydrates known as simple sugars in which generally follow the formula unit of CH2O and are the building blocks of which larger carbohydrates are made. These are the monomers of carbohydrates. Disaccharides are two monosaccharides joined by glycosidic linkage, also known as a 1-to-4 linkage, in which is covalent bond formed between two monosaccharides by a dehydration reaction. For example, there's maltose, in which is glucose and glucose combined, and lactose, in which is glucose and galactose formed together. Of these polysaccharides, there are two categories: that of storage, in which hold energy, like that of starch of plants and glycogen for animals. These are composed of alpha glucose, and then the other category is that of structural, like cellulose, in which hold the plant together through tough walls that enclose the cell, and they're made of beta glucose. The role of proteins is rather instrumental. They speed up chemical reactions, which is the main majority of them through enzymes, while others play a role in structural support, transport, storage, cellular communication, movement, and defense.
The primary structure of a protein is its unique sequence of amino acids. The coils and folds in a protein are called secondary structures, which are held together by hydrogen bonds. The beta subunits are pleated and the alpha helix subunits are coiled. The van der Waals interactions help hold together nonpolar side chains. Hydrogen bonds strengthen polar side chains and ionic strengthen positive and negative side chains. A disulfide bridge is a covalent bond that can further reinforce the tertiary structure. The overall protein structure with two polypeptide chains.
Amino acids are organic molecules possessing both carboxyl and amino groups. There are 20 amino acids in protein. Polypeptides are polymers of amino acids forged through peptide bonds, which are covalent bonds between the carboxyl group of one amino acid to the amino group of the other, formed by a dehydration reaction. When a protein denatures, it loses its native shape and hence its ability to function. This is due to pH, salt concentration, temperature, and so on. When a protein renatures is when it returns to its native shape.
This is Louis the lipid. Louis says, lipids are nonpolar hydrophobic compounds which are soluble in organic solvents. Dehydration synthesis is forming an ester linkage between the carboxyl group of a fatty acid and the hydroxyl group of an alcohol monomer, such as glycerol. Enzyme lipase is a digestive enzyme needed to digest fat. Lipase is an enzyme that hydrolyzes lipids, the ester bonds and triglycerides, to form fatty acids and glycerol. Monomers are glycerol and fatty acids, while polymers are triglyceride. Ester linkage is where the triglycerides are lipids consisting of one glycerol molecule bonded with three fatty acid molecules. The bonds between the molecules are covalent and are called ester bonds. They are formed during a condensation reaction. Hydrolysis is the breakdown of a substance by the addition of water. Fats and oils are hydrolyzed by moisture to yield glycerol and three fatty acids. Fatty acids have a bunch of carbon, hydrogen, and oxygen bonds. In saturated fat, fatty acids are in which all carbons in the hydrocarbon tail are connected by single bonds, thus maximizing the number of hydrogen atoms that are attached to the carbon skeleton. An unsaturated fat is a fatty acid possessing one or more double bonds between the carbons in the hydrocarbon tail. Such bonding reduces the number of hydrogen atoms attached to the carbon skeleton.
A fatty acid is a long chain of hydrocarbon molecules containing carboxyl acid.
A phospholipid is a lipid made up of glycerol joined by two fatty acids in a phosphate group. The hydrocarbon chains of the fatty acids act as nonpolar hydrophobic tails, while the rest of the molecule acts as polar hydrophilic head. Phospholipids form bilayers that function as biological membranes. When phospholipids are added to water, they self-assemble into double-layered aggregates bilayers that shield their hydrophobic portions from water. A hydrophilic head is having affinity for water where it likes the water, while the hydrophobic tail is excluded from the water. A steroid is a type of lipid characterized by a carbon skeleton consisting of 4 rings with various chemical groups attached. Bye, Louie. Nucleic acids, along with other polymers, are formed by dehydration synthesis. The nitrogenous bases are made out of, depending on the base, nitrogen, hydrogen, oxygen, and carbon. The sugars are made mostly out of hydrogen and oxygen, while the phosphate group stays bonded to the oxygen and the phosphate. Nucleic acids such as DNA and RNA enable living organisms to reproduce their complex components from one generation to the next. Nucleic acids are structured with a nitrogenous base and a phosphate sugar backbone. Nucleic acids are also polar due to the negative charge on the phosphate group. DNA in the nucleus programs protein production in the cytoplasm by dictating synthesis of messenger RNA in eukaryotic cells. Nucleic acids are made from monomers called nucleotides. These nucleotides are called polymers, which form DNA. Deoxyribonucleic acid is self-replicating nucleic acid which carries genetic information. Ribonucleic acid is a nucleic acid present in all living cells. Its principal role is to act as a messenger carrying instructions from DNA for controlling the synthesis of proteins, although in some viruses, RNA rather than DNA carries the genetic information. A phosphate group is a group of 4 oxygen atoms surrounding a phosphorus atom. This group can be attached to nucleotides through phosphodiester. The phosphate group links the 2 sugars of nucleotides, creating a sugar-phosphate backbone. Purine is one of the 2 families of nitrogenous bases. It's a 6-ring atom structure of rings of carbon atoms with nitrogen atoms connected to a 5-ring structure of carbons and nitrogens. Purines consist of adenines and guanines. Pyrimidine is one of the 2 families of nitrogenous bases. It's a 6-ring atom structure rings of carbon atoms with nitrogen atoms joined. Pyrimidines consist of cytosine, uracil, and thymine, otherwise known as CUT for initials. Base pairs always pair certain bases with others to form a double helix for a DNA molecule. Thymine is part of the DNA helix which bonds with adenine. Adenine is part of the DNA helix which bonds with thymine. Guanine is part of the DNA helix which bonds with cytosine, and cytosine is part of the DNA helix that bonds with guanine.